Cell division, the life cycle of the cell. You should recall much of this from introductory biology. So again, I'm going to give you the basic overview of these processes. And I have a video that goes into a little bit more detail. That's at the end of this module. So mitosis refers to the process of regular old cell division. One cell becomes two cells, each with the same amount of DNA as the original cell. Meiosis describes only the creation of gametes. The female gamete is the ovum, and the male gamete is the spermatozoa. Sorry. You can just abbreviate that to sperm. Ovum, also called an oocyte. Okay? In this process, one cell becomes two cells with the same amount of DNA as the original cell, but then they divide again so that each daughter cell has half as much DNA as your whole genome. Remember, you have a total of 46, pick a different color here, 46 chromosomes in your body. 23 from mom, 23 from dad. 23 of mom's came from her ovum, and the 23 from dad came from his sperm. When you added the 23 and the 23 together, you get 46, and that's your regular body chromosomes. When one of your regular cells divides to make another copy of itself, liver cell divides so that you have two liver cells, it's going to first double its DNA. 46 times 2 is 92. Then it'll divide again into each cell, which has the same original number of chromosomes. In meiosis, you do all of this, but then each one of those daughter cells goes on to make its own daughter cell, but without this extra doubling step that you had in the beginning. It just divides again into 23 each. If you have 23 chromosomes in a regular cell, it's incapable of dividing and it would just die. If you have 23 in an ovum or a sperm, that's made to have that many chromosomes. That's okay. Other cells wouldn't survive that way for very long. The first stage is interphase. This is not actually a phase in mitosis. It's a phase that precedes mitosis, and it refers to the cell's normal active state, just regular old going about its business, eventually getting ready to divide. During the G1 phase, the cell grows rapidly. It makes proteins. It makes more cytosol so that it can be physically bigger, so that when it divides in half, each cell isn't too small. And towards the end of this phase, your centrioles replicate. Your centrioles, if you recall, make microtubules, those long 9 and 2 protein strands of cytoskeletal architecture that do a lot of doing, a lot of building when you need to do stuff. The S phase is where DNA is replicated. The S refers to synthesis. First, the DNA is uncoiled to create a replication bubble. Separation occurs at the replication fork, so you had this twisted DNA. There's your replication fork. DNA polymerase latches on and makes a new strand of DNA based on what it reads on each strand of DNA. Finally, behind it, as it moves down that length, it retwists, it patches off gaps, and it twists the DNA back into its double helix arrangement. If you recall, when the DNA polymerase moves down the length of DNA, you have what are known as leading strands and lagging strands. On one strand, when the DNA polymerase moves its way down and makes a new strand of DNA, it can just read and build. On the other strand, as it moves down, it can't build in that same direction. So when it builds DNA, it has to build it like this and then move forward and do a little more of this and then move forward and do a little more of this building. It can only build in one direction, a 3' to 5' direction. Sorry, it reads 3 to 5, it builds 5 to 3. So at the end, these parts of the backbone need to be patched back up. Each one of these strands is called an Okazaki fragment, and this original DNA strand that you have to do this patchwork thing against is called the lagging strand. The strand that you can build DNA off of seamlessly is the leading strand. Okazaki. Let me write that here so you can see it. I've embedded this video here, which is fairly short and shows you the mechanism of DNA replication. I've also posted at the end of this module a video that shows not only all of this stuff, but with additional narration and goes into the process of mitosis as well. I strongly recommend that you watch it to fill in whatever gaps you have in this information, okay? Because Intro to Bio was a prereq, I assume that you understand this and just need a quick, broad refresher over it. But I also assume that once you get that broad refresher that you can fill in the details on your own because you're already familiar with it. So the G1 phase is basically setting the stage for cell division to happen. It's a subset of interphase towards the end of interphase. So G1, you're growing things so that when you divide, no individual daughter cell has too little stuff. The S phase specifically is about DNA replication. Once you've made more of everything else, then you know that everything's okay to make more DNA. You don't want to go into the nucleus and unwind the DNA and do stuff with it, which could potentially damage it if you don't absolutely have to. So you get everything else ready first, and then you replicate your DNA. Once you have a lot of everything else, and you have double the DNA as you need, as an ordinary cell would need, you go into G2 phase, wherein enzymes and molecules needed for cell division move to their respective sites. So G1 is growth, is about growth. S1 is about DNA synthesis. And the G2 phase is like setting the stage. Staging. Again, these are all part of the cell's life cycle preceding mitosis, but not actually part of mitosis. I like to ask this type of question on exams a lot. Hint, hint, hint. During the mitotic phase, cells are actually dividing. There's a video embedded here. Please watch it. This shows actual footage of cell division. It's got very dramatic music, and it shows very nicely chromosomes lining up, moving, doing all of their movement stuff during mitosis. Mitosis really just refers to moving. Prophase is the first phase of mitosis. During this phase, the nuclear envelope dissolves so that the DNA is loose. It's not bound up in the nucleus anymore. Centrioles make microtubules to grab on to the middle of the chromosome's X's. So part of the X phase was also the winding up of DNA into discrete chromosomes. So now you have the X's. There's your nucleus. The first part of prophase, the nucleus dissolves. Then the centrioles, which look like these little barrel guys, build microtubules to grab on to the middle of the chromosomes X's. Each one actually grabs onto more than one, but this is just easier to draw. During metaphase, the centrioles move to opposite sides of the cell, and the microtubules line the chromosomes up in the middle of the cell. So we'll do a different color for metaphase. So what'll happen is, if this is the overall huge cell, and we're going to forget about the other organelles, the centrioles move down here to either pole of the cell, and they line up the chromosomes down the middle. Remember that microtubules are constantly being rebuilt and broken down. You can build these very, very, very quickly to grow things, to push things, to move things. So what's happening here is the centrioles are making more microtubule to extend the distance between itself and the chromosome until the centriole is at one far edge of the cell and the chromosome's in the middle. During anaphase, which I'll do in blue, the X's get broken up. So they're still being held onto by your centrioles. But the X's get broken up. Oops, that one's backwards. Oh well. So now, instead of having firm X's in the middle, you have these broken in half X's. And finally, during telophase, a cleavage furrow is created. The cell splits in two. There's the X's on either side. The cell splits in two. The nuclear envelope is reformed, and those mitotic spindles made of microtubules break down. So they used to be attached, but now they're gone. Again, just to recap quickly broadly, the steps of mitosis involve breaking down the nuclear envelope so that your DNA is loose, grabbing onto the DNA with bicentrioles, moving everything so that the centrioles are at each opposite ends of the cell and the chromosomes are lined up in the middle, breaking up the chromosomes from the middle, and then splitting the cell down in half between where those X's, where those chromosomes were broken up. You pinch off that cell in between them. Now you have two different cells, so you make each of the daughter cells look like the original cell. You bind it back up in the nuclear envelope, you get rid of your microtubules. In theory, all of your organelles have been relatively evenly distributed between those two daughter cells. They had already staged themselves so that they would be evenly distributed. In cytokinesis, this is where the cleavage furrow deepens so that the cells are actually split completely in two, and the cells can move apart slightly. Telekinesis involves the pinching off and the beginning of where that sort of dotted line, where that perforation is going to be. Cytokinesis involves actual severing of the two daughter cells from one another. Finally, let's touch on apoptosis, autolysis, and autophagy. They're somewhat similar concepts, but they're not identical. Apoptosis refers to programmed cell death. Cells hit a certain point for a variety of reasons in which they're no longer useful and they will kill themselves. They'll undergo this process after one of many different things. Basically, once it gets so old or so degraded or broken down that it can't really do its job anymore, it'll start this process. Cells age by a bunch of different mechanisms. And we're familiar with all of these too, because we've heard about these things happening to us as a whole organism. Overall wear and tear. So cells have been doing a lot of things. There's a lot of metabolic byproducts that have been bouncing around doing bad things. Structures wear out. You don't have as many things to recycle anymore. Simple wear and tear. Mitochondria theory involves the aging of the mitochondria. Remember, they act in a lot of ways like separate organisms from the overall eukaryotic cell. So those start to experience their own internal wear and tear. They slow down. They can't make as much energy as they used to. And so the cell just doesn't have as much energy to work with as it once did, and it can't really do its job anymore. Another theory involves that your genes just sort of wear down. So at the end of your genes, at the end of your chromosomes, you have these extra long segments of repeating nucleotides called telomeres. And every time your cell divides, a little bit of that telomere gets chopped off. And the shorter your telomeres get, the less, well, once the telomere gets so short, it can no longer divide. It's no longer capable of cell division. Once that happens, if the cell can't divide anymore, then the only option it has left, once it's done its regular day in, day out stuff, is to die. It can't do anything else. As an organism, we also just age by all of those same reasons that you've probably heard of before, plus an immune system theory, which is sort of a variation of wear and tear, wherein the immune system, in its process of inflammatory processes and getting rid of pathogens and attacking tumors you never knew you had, and this, that, and the other thing, have done damage. The immune system does a great job at what it does, but it is a little bit of a bull in a china shop, and it does hurt us too. There's some damage to us too. So after a lifetime of fighting diseases you didn't know you were ever exposed to, getting rid of cancer cells that you never knew you even had, protecting you from all the dust and debris and the toxins and the whatever that you've consumed over your life, your immune system has been doing a lot of work, and it's roughed you up a bit. And the byproducts of it having to do a lot of work is what causes human aging at an organismal level. Destroyed cells, cells that were killed by something else, and therefore necrose, decomposed just because they were shot to shreds, will induce inflammation. Your immune system is going to be brought into an area to do cleanup. Cells who die naturally by apoptosis do not create this inflammatory response. They die in a much more gentle way, so your immune system isn't brought into the mix. The neighboring cells aren't disturbed. Two other related processes that we'll touch on in the next slide involve autolysis and autophagy. The prefix auto refers to self. So let's break down each of those root words. Auto refers to self, and lysis, what do you remember lysis refers to? Think hydrolysis. Breaking down. What about autophagy? Auto refers to self, and what does phagy mean? As a hint, remember that phagocytosis is a variation on endocytosis. Phagy is eating. Eating oneself and breaking down oneself. I'll talk about what that more specifically means in the next slide. Autophagy is a process that occurs constantly during your life. During autophagy, you're using your lysosomes. The cell is using its lysosomes to disassemble unnecessary or dysfunctional components. mRNA that you don't need proteins for anymore. Old proteins that you've lost a use for. Fatty acids that are just bouncing around and wreaking havoc. Whatever you find in the cell that you ordinarily make, but you don't need anymore. You can take it apart and recycle the parts though, which would be great. So autophagy refers to lysosomes working to break down stuff the cell has made that it no longer needs or doesn't work anymore. and then degrading those things so that it can recycle the parts. Pretty cool trick. This is a less wasteful way. It means that you have to consume less for your cells to survive. Autolysis, on the other hand, refers to self-decomposition. This is a process that happens after apoptosis. Apoptosis is where the cell's genes tell the cell, all right, you know what, it's time to die. Let's start this. Once the cell has gone through that process, the process of apoptosis, it now goes into decomposition. And it's a more orderly kind of a process than you would think of as a whole organism decomposing. When a whole organism like ourselves or animals decompose, we have autolysis happening, but more swiftly and obviously, we have other things breaking us down, bacteria and fungus and ants and whatever. This is a more gentle process, let's say. It doesn't have the help of these other entities to break itself down, and it also doesn't really disturb the neighboring cells so much. In the process of autolysis, cells release lysosomes and digestive enzymes into itself. So if you think about the lysosome in the cell, it's a bag, right? A lipid bilayer bag with breaking down stuff enzymes and acid. The inside of a lysosome is more acidic than the cytosol. During autolysis, the vesicle goes away. So you've just got loose enzymes in this little puddle of acidity that's now free to do whatever to break down and hurt whatever's immediately adjacent to it. Those enzymes break things down around it into their component parts. As the cell is broken down into component parts and is slowly dismantled by this process of autolysis, you now have all these spare parts kind of just free in the spot where the cell used to be. Because of this, the nearby cells can neutralize lysosomes with their own sort of protective mechanisms and pick up those component parts from the environment so that they don't have to make any more of their own. These parts still get to be recycled. Really cool trick. So not only is autolysis and apoptosis not damaging to the neighboring cells, it can actually help them out a little bit. If you release a bunch of nucleotides around and your cell needs, in the nearby cell needs nucleotides, cool, it can swipe it up from the dead cell. If they don't want them, your body will simply wash that away as waste. Remember, all of these cells that we're talking about in anatomy and physiology are embedded as part of tissues. Around cells, you have interstitial fluid. That interstitial fluid is basically what these things are going to get released into, because that's the environment immediately surrounding the cell. If the nearby cells can't use or don't want anything that was released during apoptosis and autolysis of a dead cell, then as the interstitial fluid is washed away back into venous drainage or the lymphatic system, it'll just get washed away out of that area. That space will be filled by one of those neighboring cells dividing.